Hello, everyone, and welcome to today's webinar on the Patchliner and the SyncroPatch 384PE. This is Cecilia Farey speaking, Marketing Director at Nanian Technologies in Munich, Germany. And we're very happy that so many have registered and signed in, especially, of course, for the night owls in Japan and the early birds on the U.S. West Coast. I would like to continue to welcome and also thank today's speakers, Dr. Rajneesh Ranjan from the Human Brain Project in Lausanne, Switzerland, who would tell us about his experiences with the Patchliner and the characterization of the complete rat KV ion channel family. After that, Dr. Alfred George and Dr. Carlos Vanoy will continue with the SyncroPatch 384PE on high-throughput EFIS annotation of human ion channel variants. Then I will sum up briefly before we move on to the Q&A session. Please remember that you can ask your questions at any time during the webinar using the chat window. And remember to send all questions to all panelists so that the application scientists sitting here with me today can see your question and then answer. All questions will be answered either during the webinar session or separately per email after the webinar. I will continue to tell you a bit about Nanian. Nanian was founded in 2002, and with the introduction of the first planar patch clamp device, the PortaPatch, Nanian has been profitable ever since. With the continuous growth and organic growth of the product portfolio, but also as a company with a dedicated and a loyal staff. So Nanian has branches in Japan, United States, and China, with fully equipped laboratories for demonstrations and support. And actually this week, we're having our user meeting in Tokyo, Japan. Nanian has developed a broad product portfolio over the years, not only in the area of automated patch clamp, but also devices for automated bilayer formation and recordings, liposome formation, the Surfer family for direct measurements of transporter protein activity, and recently also the CardioExcite for hybrid measurements of excitability and contractility of cardiomyocyte cellular networks. Returning to Nanian's automated patch clamp system, it all started out in 2003 with the introduction of the PortaPatch, recording from one cell at a time, basically replacing a conventional patch clamp rig, however, with a drastically shorted learning curve. The Patchliner was Nanian's first fully automated system, launched in 2006, and records from 8 cells in parallel. The SyncroPatch 96 came 3 years later and was Nanian's first step toward a patch clamp-based high-throughput screening platform. So what we did here is to take the heart of the SyncroPatch 96 and scale it up to 384, and there we had the patch engine, PE. So we took the patch engine and put it into a state-of-the-art liquid handler, basically to get the best out of 2 worlds, which proved to be a very successful approach since we could rely on decades of expertise in liquid handling provided by our partners and focus on what we do best, which is to develop and support patch clamp-based systems. And there the SyncroPatch 384PE was born. Just a comment on the patch substrates. All our platforms use borosilicate glass substrates for the patch clamp recordings and support high quality and stable giga-ohm seals. The webinar today will focus on the Patchliner and the SyncroPatch 384, and I'll start telling a bit about the Patchliner. It's a system ideal for medium throughput screening and ion channel research, and it allows a vast experimental freedom. For instance, a rapid solution exchange. What you see here is a dose response curve from alpha-7 nicotinic acetylcholine receptors. Another useful feature with the Patchliner when working with ligand-gated ion channels is the brief compound exposure, even down to a couple of 100 milliseconds, which is helpful since desensitization can be exposure time dependent and not only concentration dependent. The Patchliner supports two modes of temperature control, either transient temperature jumps, where the solutions are heated in the patch pipette. So this can be used, for instance, to study temperature-activated ion channels such as the TRPV family. The Patchliner also supports recordings at a physiological temperature. So in the data example here, you see High currents and the effect of erythromycin at room and physiological temperature. Speaking of High, concentration response analysis can efficiently be performed with the Patchliner. Here are examples of High inhibitors. Important in this context is that we don't see any problems with compound absorption, which is of course important in safety testing assays. The Patchliner also supports internal perfusion, which is seen here, where potassium currents are abolished after the internal administration of cesium ions. Also important to note from this data set is that the experiment was made on primary rat astrocytes. The Patchliner has shown great results with a wide variety of primary cells, and if you're more interested to learn about this, please let us know or check our website for the publication list. For the purpose also of primary cells or other scarce or expensive cells, we've developed a protocol for minimal cell usage, allowing as few as 400 cells per well, where we still obtain reasonable success rates. The Patchliner was the first platform on the market to support automated current clamp recordings. Here you see action potential from a stem cell-derived cardiomyocyte. And this also illustrates the feature of the Patchliner that each recording channel can have individual voltage and current protocol. This is important for automated, this is important for this types of experiments, but also for other assays. So the Patchliner has been quite successful in academic research, but has also been readily adopted in pharmaceutical companies and CROs around the globe. So, as I already mentioned, the patch engine is a patch clamp module capable of recording 384 cells in parallel. The patch engine fully integrates in advanced liquid handling robots from Beckman Coulter and SciBio, allowing the use of already existing hardware, software pathways for complete integration of the system into high-throughput screening environments. Important to note here is that the Biomech robot can host two patch engine modules and therefore allowing 768 parallel wells recorded with 768 individual amplifier channels. With the SyncroPatch, you can obtain a data throughput of 20,000 data points per day, generating reliable data from diverse targets. The module supports single and multi-hole substrates, which can be customized regarding resistance and the number of holes per well. As already mentioned, it integrates fully into pre-existing HTS environments. This is the Biomech platform hosting 2 patch engine modules. Each module can run through 3 plates an hour approximately, and with a single point protocol, this would allow at least 20,000 data points per day, and of course more if you make multiple additions to each well. We've also obtained expected pharmacology in HERG assays. Just an example with a couple of the usual suspects here, all well in the IC50 range reported in the literature. The success rates for completed recordings are high and ranges between 85 to above 95%, depending on the assay and if single or multi-hole substrates are being used. Just as with the Patchliner, the SyncroPatch 384 and 768 PE allows for rapid solution exchange as well as brief compound exposure. Here 2 data examples are shown, nicotinic alpha-7 responses and a dose response curve on the NMDA receptor. The accompanying software is intuitive and allows easy setup of protocols. And after the experiments, just with a few clicks, you have a report in your hand and where the data can be fed into any database of your choice. 2 features are under development. It's temperature control and automated current clamp recordings. And these will be launched later this year in Q4. The SyncroPatch 384-786 was released in 2013 and has since then been installed in academia, big pharma, and CROs around the world. And you don't have to do such exciting applications such as the PortaPatch in space. We are there for all our customers. Also, when it comes to help with assay development or tweaking HERG protocols or getting the most out of your screening assays, we are there for you. And we're happy to help. And now I would like to hand over to the first speaker, Dr. Rajneesh Ranjan from the Human Brain Project in Lausanne, Switzerland. Rajneesh has a bachelor and master's degree in computer science from Pune University, India. And in 2005, he joined the Blue Brain Project EPFL in Switzerland as a PhD student where he worked on neuronal stimulation and ion channel biophysics characterization, where he completed his PhD studies in 2011. And since then, he continued as a scientific collaborator in the same lab to conclude the work on ion channel biophysics characterization. Over to you, Rajneesh.
Thank you, Cecilia. Good afternoon, everyone. So today I'm going to talk about the work we are doing on ion channel characterization, specifically on KV ion channel family. So before I start, I would like to just give you a brief overview of our motivation, why at all we want to do that. So, excuse me. So this all started from the Blue Brain Project, where our goal is to simulate a piece of cortical column which consists of many different layers, and each layer consists of many different kind of neurons, and each neuron show different kind of electrical behavior. These electrical behaviors consist of many action potentials. And in 1952, Hodgkin and Huxley were the first one to show that these action potentials are generated because of the sodium and potassium conductances. So they were the first one who did the very first model as well as experiment to show that the there are sodium and potassium conductances are behind these action potential, and they laid the foundation for today's computational neuroscience. But the key thing here is that they actually mention just two conductances, sodium and potassium conductances. Now, in this paper we published in our lab in 2005, and by then we knew many more kind of sodium and potassium channels, but they were still not genetically based. They were like slow potassium, fast potassium, delayed potassium, and similarly for sodium channels. So we grouped them into different categories based on their kinetics or different behavior, but not based on the genes. Today we know that delayed K plus current is like So today we know that delayed K plus current is a combination of KV 2.1 and 2.2. Similarly, A-type current is KV 4.1, KV 4.2, and KV 4.3. But in computational neuroscience, we still use these generic ion channels. For example, we use slow potassium or fast potassium or A-type current. Right? Now, where do we find these ion channel models? This is the one problem each computational neuroscientist has, that there are so many different kind of ion channels, but where do we find these models? So almost all these models are getting published in ModelDB. ModelDB is a repository where every computational model, neuronal model is being published, and they contain all the ion channel models. So right now the status of ModelDB is that there are about 900 kV ion channel models, whereas none of these models are modeled for specific genetically prescribed ion channels. Rather, they represent a generic ion channels. For example, you can find a D-type potassium channel or A-type potassium channel or delayed rectifier, but you would very rarely find a model of KV 3.2 or KV 10.1. So we have not reached, from computational model point of view, that specificity where we would like to model a genetic KV 1.2 or KV 3.2. We are still in a generic ion channel which are based on their kinetics. So, and not only that, there is another problem with these database, because people who take these models from ModelDB, they change the kinetics of ion channel as per their requirement for the neuronal modeling. And the result is, in this slide you can see that A-type potassium current, one experimenter or one modeler reports one kinetics, another modeler reports another kinetics, and the third modeler takes the very same model and submits as same channel but has a completely different kinetics. So here bottom line is that ion channels do not have their identity, electrical identity. And that was one of the key problems for us, because if we were to choose which one of these channel models I'm supposed to use, we had no clue. And it is very difficult at this point in time to say that this is the genuine and real model of A-type potassium channel. So that gave us a motivation to go for the screening of potassium channels. Actually, our goal is to screen all the ion channels. But in this first stage, we have characterized potassium channels, and I'm going to talk about it. So in the Blue Brain project, or it is called now Human Brain project, we are still using these generic ion channels. So we use these 13 ion channel models which represent the generic kinetics of sodium, potassium, calcium, and calcium dependent potassium channels. So we have fast sodium, slow sodium, persistent sodium, and similarly those varieties of potassium channels. We are at this stage right now, but where do we want to go is to use the real generic, not the generic, but the genetically prescribed ion channel models. And this data we can get from single cell gene expression experiment. Suppose we have a database of 350 ion channels. All ion channel models are based on their genetic signature. and we get a single cell gene expression data from neuron, then we can combine these two and to build the more accurate model. And if we can model such a neuron, then it will have effect in, we can study neuromodulation as well as drug screening, which can have a commercial value. So with this in mind, we started this channelome project where it consists of three main phases. First is we take a clean CHO cells, we stably transfect them, and we store them for a long-term maintenance. Now, and then we use automated biophysics characterization based on Patchliner, nanion Patchliner, that we run a biophysical assay, and then we do the automated model fitting. Once these models are built and data is generated, then we would like to upload them onto a Channelpedia. Channelpedia is a resource we have been building which is like a wiki which hosts all the information about ion channels, but it is more towards providing a better and accurate model for each ion channel. So that is the overall framework in which we are working on. Now, what is the current scope for us right now? So there are about 350 ion channel genes, and out of that 135 are voltage gated ion channels, and there are about 800 publications per month in ion channel research. So in order to get overview of all this data is very difficult. So that is why we think that we need to simulate these ion channels and study neuromodulation or effect of these channels in a simulation. So currently we have about 125 inducible stable cell lines, and out of that we have cloned 95 ion channels genes, and today I'm going to talk about the 40 kV ion channel genes which we have characterized. So the ion channels which you see in the pink are the one which we have characterized, and rest of the ion channels for those which we have the cell lines, but we have not characterized them because of many reasons. For example, there are channels, two-port channel, or the channels which are open at resting potential. Those are little bit tricky. Similarly, SK and BK channels, which are calcium dependent, are little bit tricky to characterize on a planar system which works on a fluoride and calcium based solution. Now, the protocol overview, these are the different protocols we apply to study ion channels, which consist of activation, inactivation, deactivation, inactivation recovery, ramp, as well as we also apply a action potential. So we apply a shape of an action potential to record the corresponding current observed in the transfected cell line. All these data are recorded at 10 kHz except action potential, which is recorded at 50 kHz. And on the right you see the solution we use to characterize these ion channels. Now we also use a room which can change temperature between 14 to 36 degree, and our experiment setup is that we do measurement at 15 degree, 25 degree and 35 degree centigrade. Nanion also has a temperature control, but since it is active heating, so you can do experiment at 25 degree or increase it to 35 degree, but since there is no active cooling installed, hence you cannot go back to 15 degree. So we wanted to get a proper Q10 for each ion channel, and also wanted to probe how does temperature dependent temperature affects the kinetics of each ion channel. So we decided to change the temperature of whole room, which is 2.5 meter by 2.5 meter, and we looked at the kinetics of each ion channels at these three different temperatures. Now, this is the one main slide where you see the electrical identity of rat ion channels genes which were expressed in CHO at 25 degrees centigrade. So KV1 family has a very distinct and different kinetics. For example, KV 1.1 and 1.6 are very similar. KV 1.2 has an initial bump. KV 1.3 has a differential voltage dependence. KV 1.4 is fast inactivating ion channels. KV 1.7 is a very fast ion channels, which we observed that those kinetics are not reported that much in the literature. Similarly, KV 2.1 are delayed rectifier. KV 3 family has a very specific fast component at the start of their activation. Then KV4 family is inactivating ion channels, and KV 12.1 is truly non-inactivating ion channels, whereas KV5, KV6, and KV9 and KV8 are the silent channels. So this is how these channels behave if you apply activation protocol to these ion channels. Now one can ask, what happens to these ion channels if they were expressed in a different host cell line? Because these were the issue till now, saying that what happens if ion channels are expressed in different host cell line, or what happens if you express different ion channels gene. So we looked into different host cell lines. So we looked at 4 different kinetics, very slow to fast to very fast, and we expressed rat ion channel gene in CHO cell, HEX cells, and CV1 cells. So HEX cell is a human cell, CV1 is from monkey, and we saw very comparable result for all these ion channels in a different host cell line. Then we also looked at the across species for these ion channels. So KV 1.1 looks very same for rat, mouse, and human. Similarly for KV 1.4 and KV 2.1 and KV 10.1. So, and there is a reason behind it. So if we look at the homology report for these protein similarity between rat and mouse and rat and human, you see that they are more than 90% homolog. And we are also looking at some ion channels which are less than 90%. For example, KV 1.5, which is marked as 87.5. So these ion channels, now we are also creating a cell line to compare what happens to the ion channels which are less than 90% homolog. But here, take-home message is that these ion channels are very conserved across the species, and we see very same kinetics across rat, mouse, and human genes. And that gives another argument that if you are studying rat ion channel or mouse ion channels, it can be extrapolated to human ion channels. So now I'm coming back to the biophysical characterization, and here is the data which I would like to show for KV 1.1, KV 1.2, and KV 1.3. So at 15 degree, KV 1.1 does not inactivate, but it really inactivates at 35 degree, which is close to physiological temperature. And this is not reported that well in the literature. So many places in the modeling, I will show you one slide where people have been modeling KV 1.1 as a non-inactivating ion channel. And when you look at the references, you see that those experiments were done at the room temperature, probably with the cold solution, and that is why they don't see real inactivation. But when you look at when you control temperature properly, like we do in our experiment, you see a very systematic difference between 15 degree, 25 degree, and 35 degree. Similarly, KV 1.2 has a distinct bump at the start, and that feature is preserved even at 25 and 35 degree. Similarly, you can see KV 1.3, KV 1.4, KV 1.5, KV 1.6. So they have a huge effect of temperature on these ion channels. So KV 1.7, here you see that there are only 21 articles published till now, and it is one of the most fastest ion channel I have seen for potassium family. Now there are, for example, 554 KV 2.1 ion channel publications you can find, but there are only 21 publication for KV 1.7. One argument I could say is that we find more disease related to one channel, probably because we are studying the channel that many times. So that is another goal for this project, is to give equal attention to all these ion channels, and we make model for each ion channels, and then we use them for our simulation in a new cortical column. So as you see, so this is the KV3.1 family, KV3 family, then KV4 family, and KV6 family. So KV5, those silent ion channels, they remain silent at 15 degree and 25 and 35 degree. So now I would like to give you a quick look about the modeling. This is one example I can show for KV 1.1. So first panel shows the experimental data which we have observed through Patchliner. So 15 degree trace, you see there is no inactivation, and then 25 there is some inactivation, and 35 is a very strong inactivation, you can see for KV 1.1. Now in model DB you find KV 1.1 model as a non-inactivating ion channel. So below 2 traces, they represent KV 1.1 model, and in reality they do take care of temperature control, but because their reference is experimental data where experiments were not done at different temperature, they do take account of temperature control by just adjusting the faster activation time constant, which you see in the last panel. So 15 degree is slower than 25, which is slower than 35, and this is one of such models which are being used in neuronal modeling. From this, we want to use our data, and then we want to build a model which mimics really the experimental data. So this is where we want to go, and we are in this process, and very soon we will be publishing our article with all the data. Now, all these models and data, we would like to make them available on Channelpedia. Channelpedia is a shared resource where we would like to put all the information. So it is like a wiki where if you are a contributor, you can edit any sections, and then you can add the references or useful information to this page. So I would explain you the summary of this ion channel resource in this movie, where you see different sections, for example, kinetics or experimental data. Currently, it contains the basic model and their figures attached to it. Now, in this page, in future, you will have all the experimental data available, which we have recorded till now. Here, for example, you see for KV 1.4, you see the data for 25 degrees centigrade. When you click on that, you can get all the information about what protocol was applied, how many times it was applied, so which you can browse, and if you see, you can see the action potential response here also. So once you have this, then you can also click on actual protocols which was applied to these cells, and then you can also browse in a different interface where you directly choose specific ion channel, look at how many different species data they have, you can select the temperature, and then you can select the protocol, and then you will see all the traces which we have right now. So for example, for rat ion channels, we have recorded at 3 different temperatures, and this is the data you will be seeing on Channelpedia. This data is still not available. As soon as we will publish the article, all this data would become available to everyone. Now, you would be able to download this data in different formats. For example, we would be providing MATLAB and HDF5 file format along with a function to access these data as well as plot these data. So this is an example where I'm downloading data from a cell which has an ID 736, which now can be accessed through MATLAB by a simple API. For example, a function called plot cell by ID, where you specify the ID of the cell, the protocol which you want to plot, for example KV activation, and suppose you want to plot the first repetition, and it will give you the first repetition of the cell. Then similarly, you can plot the deactivation or inactivation or KV ramp. So any recording traces which I have recorded till now, you would be able to download and look at them with the help of this data file as well as the scripts. Now each cell has an ID. Now if you want to download them in bulk, these IDs, these web addresses or download links are based on REST API. So you can just write the cell ID on the top at the end and you will have access to the cell which has an ID 7000, for example, in this case. Okay, so this is the overall functionality we will be providing within 1 to 2 months. We are still in the process of writing these data. So apart from this, we observed there are some peculiar features for each ion channel. For example, KV 1.3, 1.5, 3.3 and 3.4 and KV 10.1, we found they were very variable in nature. For example, if you record from one cell, you see one response, then another cell has a slightly different response. So that brought us a question that if each cell is behaving a little bit differently, then how can we talk about identity? But we looked at the genetic aspect of it and the reason behind the variability, and we think that we have the answer why these ion channels are different or variable in nature. So that result also we will be publishing with our article. And then another thing I wanted to share with you is result on KV 7.1 temperature sensitivity. So there are about more than 1,000 articles available on PubMed for KV 7.1, which is involved in cardiac arrest and LQT syndrome, atrial fibrillation, etc. And this ion channel, if you record at 15 degree, you see a very nice current, significant current and signal to noise ratio. But the very same cell, if you increase the temperature from 25 to 35 or even at 25, you see very less current, and that disappears at 35. My question to ion channel community and people who are using these channels for drug screening is that if you screen them at 25 and actually it is not conducting any current at 35 degree, then how can we be so sure about our screening method? There are many such observations we have observed for many different ion channels, which also we will be listing as in our discussion for our publication. But for the time being, these are the main things which I wanted to share with you. So to conclude my talk, we have isogenic inducible cell lines for entire KV family or potassium ion channel family. We did a standard biophysical characterization of the complete rat KV ion channel family, and data will be made available via Channelpedia. Each KV ion channel has a unique electrical identity, and electrical identity can be translated to human gene. KV ion channel kinetics is very sensitive to temperature, and there is a possibility to assess effect of any mutations on ion channel kinetics, which we have not shown today. Now in future we want to look at KIR channels, 2-port channels, HCN, BK channels. All are cloned, but they have to be recorded in a properly adjusted condition, and their assays has yet to be developed. And then we will move to sodium potassium TRP channels as well as heteromers and beta subunits or co-expression system. Now I would like to thank Emmanuel, Michaela, Mimi and Valerie and Dayan who helped me in a respective manner, and also the funding agency, which is EPFL Blue Brain Project Fund, as well as the ETH Board of Funding to the Blue Brain Project. Thank you all, and I return the control back to Cecilia, and I'm looking forward for your questions. Thank you.
Thank you very much, Rajneesh. And we now move on to our next two speakers. Okay. So now it's time for our speakers from the Northwestern University Feinberg School of Medicine in Chicago, Illinois. Welcome Dr. Alfred George and Dr. Carlos Vanoy. Dr. George received his medical education at the University of Rochester and then trained in internal medicine at Vanderbilt University and in nephrology at the University of Pennsylvania. Dr. George joined the faculty of Vanderbilt University in '92 and was promoted to the rank of full professor 6 years later. In '99, he established the Division of Genetic Medicine and served as division chief until 2014. Currently, Dr. George is Magister Professor and Chair of the Department of Pharmacology and Director of the Center of Pharmacogenomics at the Northwestern University Feinberg School of Medicine. Dr. Carlos Vanoy received his undergrad degrees at Texas A&M University, where he worked for 5 years as research assistant before joining the PhD program in the cellular physiology and molecular biophysics at the University of Texas Medical West at Galveston. In 2002, he joined the research group of Al George in the Division of Genetic Medicine at Vanderbilt University. And in 2014, he moved to the Department of Pharmacology at Northwestern University, Chicago, Illinois. So I hand over to you, Al and Carlos.
Thank you, Cecilia. We're using a single telephone, so with me is Dr. Vanoy, and I'll be doing the primary presentation, but we'll call on him randomly to give his input into some of the experimental data, which is really all his doing. We're going to talk about our work using the SyncroPatch 384 platform that we've had for about 9 months now. We initially purchased this back and had it installed back in October last year. Our primary motivation was twofold. In our new Department of Pharmacology at Northwestern University School of Medicine, we wanted to set up a high-throughput screening facility that was targeting molecular targets that could be assayed electrophysiologically, such as voltage-gated ion channels. And secondarily, a big part of our research program over the years has been to elucidate the functional consequences of ion channel mutations, human ion channel mutations associated with channelopathies.
Al, sorry to interrupt,
yes.
But you need to share your presentation.
Well, I haven't really said much yet, so you haven't missed anything. I just had. Why don't we do that then? So let's see. How's that?
That's perfect. Thanks.
Yeah, I'm just rambling about the title slide, so I haven't really.
Okay. Go on.
Thank you for letting me know.
Yeah, sure.
So anyway, our second motivation was to have a high-throughput platform with which we could do a large-scale functional annotation of human ion channel variants associated with inherited disorders, such as the one I'll primarily talk about today, which is the congenital long QT syndrome, as well as certain forms of epilepsy. And although a large part of our work on the SyncroPatch has been devoted to optimizing protocols for measuring sodium currents, we've also done quite a bit of work on potassium currents, and I'll focus our presentation on the potassium channel studies that we've done so far. So just sort of the broad picture here is our main focus has been on channelopathies, and over the years we've used genetic paradigms to identify mutations in various ion channels, such as this illustrative topology of a voltage-gated sodium channel. We've then turned, as many labs around the world have done, manual patch clamp recording in heterologous cell systems to ascertain or elucidate the functional consequences of specific genetic variants, and then to try to understand how that explains the disease biology. And the long-term goal is to ultimately use that information to possibly model in a dish the disease and use the abnormal functional data that we've collected as a way of assessing whether there is a pharmacological opportunity, and to potentially use pharmacological profiling of existing drugs or possibly look for novel matter to correct the problem. With manual patch clamp, this has been an effective way of approaching the problem back when there was relatively few mutations known. But now, with really the explosion of next-generation sequencing and a large number of commercial genetic labs testing many hundreds, if not thousands, of subjects who have genetic diseases that could be channelopathies, there is now thousands of mutations that are known, and there's a real divide between what we know about the functional effects of individual mutations and how many different mutations there are. So this big challenge of assessing the pathogenicity of variations in ion channels has really become where manual patch clamp has really become a huge bottleneck. And just as this slide illustrates, we can have a whole lab of people doing patch clamp all day long, 5-6 days a week, and on a good day, a good person, good postdoc will get to keep about 16 good measurements per person per day. And over the course of a lifetime of a postdoc, he might be able to characterize a small number, a few dozen mutations over the course of a few years. So it's just really a low throughput approach. So that was one of our major motivations for investing in automated patch clamp. And we assessed really the two major 384 well systems that were on the market at the time last year. The Nanion system is what we decided to invest in, and the measurements, and the reasons for that are illustrated on the next slide, which were we felt that among competitor systems, we were able to see a much higher rate of giga-seal formation on the SyncroPatch. There was available series resistance compensation and leak subtraction, things that we were used to doing with manual patch clamp. We also were very happy with the seal stability that was illustrated to us, and we thought that would enable us to do multiple voltage protocols in series, just like we would do normally on manual patch clamp. Although we bought a 384 well system, we're looking very seriously expanding that to the 684 platform with 2 modules, and looking forward to the developments of that you heard about earlier in implementing current clamp as well as voltage clamp. We were really pleased that the platform runs on a Biomech FX robot platform, which we're just a couple of hour drive from the Biomech headquarters in Indianapolis, which means we have excellent service in our area. And we also liked several other features about the robotic handling. We also had worked at our time at Vanderbilt, worked extensively with the Patchliner and have data in a couple of publications. And so we were very familiar with the operation and the support from Nanion, and so those were all very positive influences. We did know that there were some limitations in the platform that are works in progress, of course, but we, in doing sodium channel recordings, we were aware of the limitation that the smallest recording segment was 10 milliseconds, and that did limit some of the experiments we would do, such as recovery from an activation of fast-gating sodium channels. There is also some limitations that we understand are hardware limitations, that the number of individual recording segments that can be run is limited to 64. That limits us to the number of pulses we can throw at a sodium channel in a use-dependent experiment. So far, I will have to say that it hasn't really been a major limitation for our work with the cardiac sodium channel, and so far with the neuronal sodium channel, we haven't really pushed it. But I think at some point there are some potential experiments that we would prefer to do with a larger number of pulses. And then, although, as you've heard, the current instrument does not have active temperature control as available on the Patchliner, but there's now going to be a solution for that in the near future, as there will be for other things that I've mentioned, and we're looking forward to the next software update, which will include an ability to go down to 1 millisecond recording intervals, and the hardware limitation is still a work in progress, and as we heard, temperature control is something that can be modified now. So I'll focus the rest of my talk on on 3 different potassium channels, casein Q1, casein H2, both of which are long QT-associated genes in humans, and lastly, a neuronal channel, KV2.1, which we've recently identified as a novel epilepsy-associated gene, and give you some vignettes about how these channels are behaving on the SyncroPatch for us. Casein Q1 is the major gene associated with long QT syndrome in humans. It is responsible for about 50% of the known mutations. There are now in the human gene mutation database over 500 mutations in long QT syndrome, and we know for sure that genetic testing companies, commercial genetic testing companies, have many more mutations in their databases. We've in fact worked very closely with one of those companies, and they've shared with us several dozen variants of unknown significance, which has become a major problem in this disease as well as most genetic diseases, in which there are variants that are detected in a patient with the disease of interest, but for which that mutation can't really be well classified as to its pathogenicity for a variety of reasons. Either it's not been previously described in association with the disease, or there's no supporting biological data to support its pathogenicity. And for an ion channel, it's relatively straightforward, as we know, to perform a functional study to ascertain its functional consequences and then to infer pathogenicity by those data. So we have really set out to do a large-scale functional annotation of casein Q1 variants in a project that is now funded by the NIH. We're looking to study well over 100, probably around 140, 150 variants over the next few years, and be able to then build a computational predictive model that can be data trained with our experimental evidence of pathogenicity, and then try to make a predictive algorithm that's very accurate for the next mutations. So we've casein Q1 is one component of the slow delayed rectifier current in the heart that's essential for repolarization, and the mutations that impair this current cause long QT syndrome. Casein Q1 doesn't function alone in native tissues, but rather it partners with its accessory subunit casein E1 to make this slow delayed rectifier current that's illustrated on this slide. We initially set course down the pathway of making stable cell lines on every single mutation that we were planning to study on the SyncroPatch. Fortunately, I recently hired a former member of my lab who had spent a few years in the pharmaceutical industry, and she showed up and said, well, I'm not sure you need to make stable cell lines. You can use electroporation using this MaxCyte system that we used to use at our company, and we've since purchased that system and have integrated that into our workflow with the SyncroPatch. So the data that I will tell you about today is largely that of transient transfection of CHO cells with casein Q1 or casein Q1 plus casein E1 and recordings on the SyncroPatch. So as an example, this was one of the first experiments we did to test the feasibility of using the MaxCyte electroporation system to co-transfect CHO cells to express transiently casein Q1 and casein E1, both of which are of human origin. This is, again, one of the early experiments, and on this experiment, we had more than 73% of cells with a giga-seal or better, and the current properties were very reminiscent of what we had seen and others have seen with manual patch clamp. And then a more direct comparison of manual patch clamp here shown on the left with SyncroPatch recordings on the right showed very similar kinetics, albeit slightly lower current density with cells recorded on the SyncroPatch. And we think this is due to the fact that when we're doing manual patch clamp recording, we co-transfect with a co-transfection marker such as GFP, and then look for cells that have the most prominent brightness. And therefore, we're consciously visually selecting cells that probably on average have greater expression levels of the plasmids. So we think that's a very good possibility. Whereas with the SyncroPatch, we're not selecting cells visually, and therefore there's a little more random selection, and probably it's a population effect. If we normalize to the maximum current recorded at the most positive test potential, we can actually see that the kinetics are virtually identical, and so we're getting really good, accurate representation of IKS on the SyncroPatch. Here's some additional analyzed data, which includes on the left a current voltage relationship comparing, again, manual patch with SyncroPatch. Although the numbers of cells that are interrogated is much, much higher with SyncroPatch, the overall shape of the curves are very similar, albeit lower average current density on the SyncroPatch. And again, we think this is due to selection bias when we do manual patch. And then the plot on the right is the activation curve. That's essentially normalized tail currents. Tail currents, right? Tail currents from which we can deduce the voltage dependence of activation. And Boltzmann fits of these data show there's really no major difference between manual patch and SyncroPatch. Most of our experiments that we've now done to look at variance are doing casein Q1 alone versus casein Q1 plus casein E1. And here I just chose to just show you some examples of the casein Q1 alone data to give you a flavor for what we're doing. So this is, again, a pretty relatively typical experiment. Maybe it's a little better than average, but here we, again, use the MaxCyte electroporation system to transiently express wild-type casein Q1 in CHO cells, and then place those cells on the SyncroPatch. A couple of just additional experimental details. The cells are electroporated on day 1. We grow them for 2 days, and then either directly move them to 3 days, and we either directly move them to the SyncroPatch after 72 hours, or after 48 hours, we can freeze the cells and then recover them later when we're ready to do the recording, grow them for 1 more day, and then place them on the SyncroPatch. And so we've done it both ways. I think this experiment was probably done post-frozen. So these cells were transfected, grown for 2 days, frozen, and then recovered on a separate day, grown 1 day, probably overnight at 28 degrees to try to boost expression, and then placed on the SyncroPatch. And so here we have an example of wild-type casein Q1 in which 83% of the cells had at least a half a gigohm seal or better. Thank you. Now we've used this system now to look at a large number of mutations, and I think plotted here is just the current density from CHO cells that were expressing one of 28 different variants that come from various sources, some of which come from a genetic testing company called GeneDx. Others are published variants. Others are from other public databases, and we've just sort of thrown in a lot of things to sort of get a contrast here. And you can see there is quite a variability. Some of the variants that have near normal function or maybe even hyperfunction actually come from either their normal variants in the general population, or some of them have actually been published and ascribed as pathogenic mutations. Whether that's true or not, we're going to keep investigating. But just to sort of, for perspective, to look at this many variants with the high density of data that we were able to collect would take a postdoc probably at least a year. And these experiments were probably completed within the span of about 3 weeks. Moving on from casein Q1 and to really just highlight a couple of other potassium channels that we've studied on the SyncroPatch. Casein H2, which encodes the major subunit that is responsible for the rapid delayed rectifier current in heart, known as IKR. Casein H2 encodes the HERG, or human erg potassium channel, that is well known. Some investigators have thought that the hERG channel requires an accessory subunit, whereas others are not so sure about that. We've actually recorded now on the SyncroPatch using a stable cell line, as again stably expressed hERG in human hERG in CHO cells, and compared those findings with manual patch on the same cell line. So this again was an early experiment we did probably within a couple of months of receiving the SyncroPatch last year. This is a stable hERG cell line in CHO cells. Here we have almost 75% of cells have near gig-ohm seals or better, and comparing the data with manual patch clamp, again we see some divergence in overall current density, which again I think we ascribed this to the selection bias for high expressors with manual patch clamp. But again, the voltage dependence of activation inferred from the tail currents is no difference between manual and SyncroPatch. And then lastly, I just want to share with you a neuronal potassium channel, KV2.1. Last year, Dr. Jennifer Kearney, my associate here in the department, and us published a study showing mutations in patients with a severe form of childhood onset epilepsy that were due to a previously unknown gene, KCNB1, which encodes the neuronal delayed rectifier current KV2.1. And in the 3 mutations that we reported in this Annals of Neurology paper last year, the 3 mutations that we identified were all within the pore domain, and by manual patch clamp, these mutations all disrupted ion selectivity, making the channel sort of non-selective for sodium or potassium over sodium. And so we're turned back to this channel on the SyncroPatch really to enable us a higher throughput system, because now there are many more mutations to study, and also this could be a pharmacological target that could give rise to new therapies in epilepsy. And so here we again go back to transient transfection in CHO cells. And so what I'm illustrating here is an example of our recent work on KV2.1 transiently expressed in CHO cells and recorded on the SyncroPatch. And again, in the lower left corner, you can see that the statistics of this experiment show nearly 74% of wells had at least half a gig-ohm seal or better. And comparing and these are room temperature recordings, but comparing the results between manual patch clamp and SyncroPatch, we see very little difference. Here I just illustrated on the right, this is the conductance voltage curve for the data collected from the SyncroPatch, and in the small type is KCNB1 wild type, both on manual patch and SyncroPatch. The activation curves really overlie, have almost identical V1/2 values. So again, we're very comfortable with the accuracy at which we're getting biophysical properties among these 3 potassium channels, and feel that we're getting very accurate recording data. So anyway, that's all I had to say. I just want to show another picture of Carlos Venoye, who's done really most of the work here, and he's assisted by Reshma Desai, who gets a lot of credit for educating us about the MaxSite system that was used prior to her coming here. And we have grants from the NIH and also from our home institution to fund this work. So anyway, thank you for your attention, and I'll turn it back over to Cecilia.
Thank you very much, Al. Very nice presentation. So I'll just make a brief wrap up here. As we've seen from both presentations now, the bottom line is that the Patchliner and the SyncroPatch 384PE generate high quality data and has an immense versatility and experimental freedom that enables the user to do screening as well as intriguing science. And for those of you attending today, if you would like to see any of our systems in action, just contact us and you can visit us for live demonstrations. So just briefly, the future outlook of the SyncroPatch 384 768 PE. There's a temperature control coming up in Q4, as well as automated current clamp recording. As you already mentioned, Al, there will be a software release allowing shorter pulse protocol segments, allowing faster frequency dependence investigations. And there will also be a software integration with gene data, a powerful tool for multiplate analysis and down to raw data traces. Right, so we'll move over to the Q&A session. So there's a question for you, Rajneesh. How do you take into account the branched shape of neurons for your model when you are recording from round cells?
Yeah, so in modeling, neuronal modeling is a little bit different than, so right now what we are doing is we are just measuring ion channel kinetics. Then we use these kinetics in a same morphology like a neuron. So there we distribute ion channel with known densities around the branches. So there we do have 3D morphology of each neuron, and there ion channels are inserted as if in real physiology you have ion channels on the membrane. So our genetic algorithm that searches for the distribution or density of these ion channels across the branches. For example, IH ion channels, they are distributed exponentially from soma towards apical dendrite. So these kind of distribution we set up, and then genetic algorithm searches a real number, actual values, so that it fits the proper recorded current traces, voltage traces recorded from the real neuron. But those were based on the ion channel models which are built on CHO cell lines. So once these ion channel models are present, we don't change their kinetics. The only thing we play with is the density of these ion channels and where they should be distributed on the branches of neurons. So that is how we build neuron models.
All right, thank you very much. Then a question for Nanian. With regards to Rajneesh's presentation, what is your experience with calcium-regulated channels? I would like to hand over to my colleague, senior scientist Nadine Becker.
Yeah, so we have developed assays on both instruments, the Patchliner and the SyncroPatch, for calcium-regulated channels, and we have assays readily available for SK channels 1 to 4, as well as for BK channels and also TMEM16A. So if anyone is interested in those protocols, we're happy to provide the solution recipes and recording protocols for those.
Thank you very much. Then a question for Al and Carlos. What biological properties are you able to test using this equipment?
The biophysical properties that we're able to look at right now, it depends on if we're looking at sodium channels or potassium channels, but common properties are the voltage dependence of activation, voltage dependence of inactivation. We can look at use dependence, specifically for sodium channels, sodium recovery from fast inactivation. We can also look at use dependence in a slow inactivation, and we have also been able to measure persistent current, which is very important for sodium channels that are involved with epilepsy, since some of these mutations cause persistent current. We were able to detect 3% levels of persistent current in our with our experiments.
All right, thank you very much. And then another question for you, Regarding the transient transfection, how do the success rates compare using frozen stock compared with using the cell directly after the transient transfection with the MAC site?
With us looking at currently to chill cells and HEK cells using either sodium or potassium channels, we really haven't seen any difference between cells used 72 hours post-electroporation or the cells that were frozen 48 hours post-electroporation and then thaw 1 day, grown for 1 day and then tested. We have not seen any difference. Anything,
All right, I see. And here another question. What do you mean with, quote, using multiple internal solutions, end quote? Do you mean that you can have different solutions across a plate or internal exchange for every cell?
Yeah, I think there was a point I made on the slide. I think the fact that we can, use multiple internal solutions across the plate, but also we have successfully changed internal solutions during recording. We've not done that very many times, but we have demonstrated as a proof of principle that we can do that. And both of those features were attractive features of the SyncroPatch that helped drive us in that direction.
All right, thanks very much, Al. Rajneesh, another question for you. Do you know if the same brilliant work that you presented is currently done for cardiac actional potential modeling, effect of temperature on biophysical parameters of cardiac ion channels?
Actually, I am not aware of any such way to characterize all possible ion channels in such a extensive manner. So there is a lot of work done for cardiac action potentials, and that is a kind of easier also because it is a little bit wider. There are a lot of models, but not in my knowledge. Again, they are not based on genetic ion channels. They are based on like known key ion channel models rather than really looking at the genetic basis for each ion channel. So that I am not aware of, but I might be wrong.
Okay. I think that's all regarding questions. So I would like to again thank our speakers very much. Thank you so much for your excellent presentations. And I would also like to thank everyone who attended today. We will send you an email with the link to this webinar and a document summarizing the Q&A, the questions that came up during the Q&A session. So thank you very much, everyone, and bye-bye.